Our genes are capable of making over 20,000 different proteins. Not all of these proteins are synthesized in every cell, however. Some proteins are only made in certain types of cell in our bodies, and that's what makes cells different from each other and gives them their special characteristics. Some proteins are made in most cells, though. We call these housekeeping proteins, mainly because they are responsible for overseeing basic processes that are common to most cells. One of these processes is the control of the cell cycle. At any point in its normal life, a cell may be in a growing, dividing, or resting state. The passage of the cell from each of these states into the others is called the cell cycle. The control of the cell cycle is crucial for the correct functioning of cells and tissues within the body and the maintenance of the appropriate number of cells in an organ or tissue. A relatively small number of genes code for a highly important group of proteins. These respond to various internal and external signals that determine what state the cell exists in at any one moment in time. If a cell is instructed to undergo cell division, it will first enter a phase called G1, where G stands for gap. This phase may last for several hours, during which the cell will increase in size and the internal components, or organelles, are duplicated. The cell will then enter the S phase, where S stands for synthesis. During this phase, which can last for over 8 hours, DNA synthesis takes place and each chromosome is duplicated. At the end of S phase, the cell will enter a final phase of rapid growth called G2, during which a large amount of specific protein synthesis takes place. These proteins are required for mitosis, the M phase, during which the duplicated chromosomes separate and segregate into the two daughter cells. If the signals for growth are still present, the daughter cells will then enter another round of the cell cycle. If not, the daughter cells will enter a resting phase, sometimes called quiescence. At certain stages of the cell cycle, there are checkpoints at which the cell makes the crucial decision whether to proceed or not. This decision depends on the presence and activity of certain proteins known as cell cycle regulators. One such protein is encoded by a gene called RB, which stands for retinoblastoma, a type of cancer found in the retina of the eye. The RB protein is able to inhibit the transition from G1 to S phase when it is activated. However, in retinoblastoma tumours and a variety of other human tumours, the RB protein is inactive, often because of mutations in its gene. This means that an important brake used by the cell to restrict cell division is lost. The RB gene is therefore a good example of the relatively small subset of housekeeping genes whose protein products are often defective in cancer cells. Tumours often have mutations in non-housekeeping genes too.